Hello everyone. Welcome to the Charting the Brain's Pain Map, Why Mine is Different Than Yours webinar. We're glad to have you here with us today. My name is Tanya Hyde and I'll be moderating the webinar today. I'm the project manager with the Canadian Institute for the Relief of Pain and Disability, and this webinar is made possible through the support of the Community Gaming Grant Program, through the BC government, as well as through donations and memberships to CIRPD. As I mentioned, my name is Tanya, and I'll be moderating the webinar and the Q&A session today. And our presenter is Dr. Karen Deborah Davis. Dr. Davis is a professor at the University of Toronto and head of the Division of Brain Imaging and Behavior Systems Neuroscience at the Krembil Research Institute. She trained as a single-cell electrophysiologist and then pioneered brain imaging to study pain. Her lab combines behavioral testing, structural and functional MRI to examine chronic pain, pain-attention interactions, plasticity, disease vulnerability, treatment effects, and concussion. Dr. Davis's work has culminated in over 175 published papers and book chapters, and she's given more than 160 invited lectures and created a TED-Ed video, How Does Your Brain Respond to Pain?, viewed nearly 1 million times. We're very excited to have her with us today. Karen, I will hand it over to you.
Great. Thank you, Tanya. Well, it's really a pleasure for me to deliver this webinar today, and I thank the CIRPD for inviting me to give this webinar and to have this audience both in real time online. Welcome everybody, and to those who will be joining us offline anytime in the future. So as Tanya mentioned, the title of my webinar is Charting the Brain's Pain Map, Why Mine is Different Than Yours. So just to set the stage, let me just begin, see if I get this going. Okay. Basically, issue that you probably are all aware of, and that is the personal and societal cost of chronic pain. But let me just give you some numbers to set the stage. When these numbers came out of both studies in the United States and in Canada, they really quite shocked me that the numbers of Americans and Canadians suffering from chronic pain are really more than those suffering from heart disease, cancer, and diabetes combined. So those numbers are quite staggering, and that translates into an enormous annual cost in the U.S. of over $600 billion and in Canada of over $40 billion that are directed towards health care, lost wages, and productivity. So clearly there are enormous costs, financial costs of chronic pain. But what I find personally the most important statistic to come out is that many people with chronic pain experience the worst quality of life compared to other chronic diseases. And this is what motivates myself and others in the field to try to work towards finding a solution to chronic pain. So when we talk about chronic pain, as I mentioned in the previous slide, we're not just talking about the pain, but we're talking about what happens to the person as a whole, and this really encompasses the field of suffering. And this can be quite staggering. And in this country, we've had quite a discussion over the last year or two around the topic of physician-assisted suicide to the level of the Supreme Court of Canada. And that's because the consequence in some extreme cases of chronic pain is that the suffering that people are experiencing is so severe that they want to end their life. They don't want to live anymore. So what can we do to change that situation, and why in fact do we even have that situation? So this is really the very difficult situation that many healthcare providers are put in, and that is a patient with chronic pain comes into their clinic and they have a number of solutions that they can offer them. So for instance, there are a host of different psychological type of treatments such as cognitive behavioral therapy. There are of course numerous drugs that are out there that can in some cases provide quite good pain relief, but not in all cases. And we also have a number of surgeries and interventions, many developed only in the last 10 years, such as deep brain stimulation and transcutaneous magnetic stimulation. So we have a lot of approaches to treat chronic pain. So what's the problem, and why are people still suffering? Well, the truth is that although many people do get a lot of benefit from these different approaches, many people don't benefit from these treatments, and some people who may benefit have such severe side effects that they can't continue using that particular treatment. So this is really the problem. Now, how do we try to figure out why some people actually benefit from one or more of these approaches and other people don't? So this is one thing that we need to address. And what we've been doing in our lab to address this question is we don't only look at chronic pain, but we're also trying to understand acute pain. And we're trying to understand the variability in acute pain sensitivity. So let's take, for an example, something in everyday life. So around this time of year, a lot of people are getting their flu shots. And perhaps you have stood in line waiting for a flu shot and saw people like this who were getting the flu shot and actually responded quite well. You know, they were pretty happy about it, didn't experience very much pain. But then there are other people that you might have noticed that actually have quite severe reactions to the exact same stimulus. So the exact same needle, same injection, yet they're responding quite differently and are clearly distressed and suffering. So even to an everyday, somewhat mild stimulus, the response of people in the general public can be quite variable. So what underlies this variability in pain sensitivity and pain experience? Clearly, it must lie in the brain. So the brains of these individuals must be different because they're experiencing the same stimulus in a different way. So how can we understand the difference in these different brains? Because really, that really lies at the heart of why these different treatments are beneficial in some people and not beneficial in others. So what we're trying to do in our lab, and other people in the field are also trying to figure out how to treat individual brains. Okay, so how do we do that? Well, the first thing is we have to figure out what is the code that underlies pain that lies in the brain. And if we can figure that out, then we can work towards trying to change it for people who are experiencing chronic pain or to treat it. So let me then begin. I just want to kind of get everybody on the same page with some very basic terminology, because there's a lot of misconceptions about some of the terminology in the pain field. So let's start with just the basic term pain, which is quite simple but is sometimes misinterpreted. So the basic understanding of the term pain is that it's something that you feel. So something that you feel is subjective, meaning only you can really say what you're feeling. And usually pain is quantified by an individual telling the healthcare provider or the researcher what they're feeling. So it's something called a self-report. And there's various different scales that can be used. Of course, the simplest scale is just, you know, a 0 to 10 scale or 0 to 100 scale that people are asked to rate their pain on. Now, the other thing, though, to be very clear about is that pain can arise in many different ways. So it's not always something that arises from a stimulus to the external body, right? So you can feel pain when you have something going on inside your body, in your muscles, in your organs, but also it can arise when something happens in your brain. So for instance, if you have a stroke, there are people that have what's called post-stroke pain, which arises because of damage to the brain itself. And in the case of people who have amputations, many of them, most of them, have what's called phantom pain, which is pain felt to arise from the limb that they no longer have. And the mechanism of that type of pain is thought to include mechanisms within the brain. Okay, so pain is something you feel, and it's subjective, and it can arise both from stimuli applied to the body and things that happen within the body. Now we have this other term called nociception, and this is something that you can actually quantify, and it's thought to be an objective measure of pain, or kind of a proxy for pain, but it's not equivalent to pain, and it's very important to understand the distinction between pain, which is something only known to the person feeling it, and nociception, which is something that you can objectively measure. So for instance, you can measure brain activity or the outcome of brain activity or something related to brain activity. For instance, we're going to be talking about brain imaging in a moment. And this is really what nociception is all about. So it's something that happens in relation to the pain, but is not equivalent to the pain. And to understand a little bit more about the distinction between pain and nociception, just think about what happens when somebody is under anesthesia. So when somebody's under anesthesia, if a noxious stimulus, or a stimulus which normally produces pain, is applied to their skin, you can still measure that response in the brain. The brain is still active, but the person isn't actually experiencing any pain because they're under anesthesia. So in this case, you can have nociception, which is the brain activity, without pain. So there are these situations where these two things become disconnected. But in most instances, measuring nociception gives you some insight into pain, and this is why we are developing, and many people are developing, objective measures of nociception. Okay. So what are these objective measures of nociception? How can we assess the brain? I'm going to give you a little bit of an overview of the technologies that are used in my lab and other people's labs to assess the brain. So first I'm going to mention various ways that we can look at communication highways and activity centers in the brain. This is kind of how I like to think about what's called the white matter and gray matter in the brain. So in the brain you have areas where there's cells, and this is called the gray matter, and that's where all the activity happens. Then you have what's called white matter, which is kind of like the highways, which connect these different regions of gray matter. So the white matter brings information from one area of the brain to the other. So there are methods that we can use to look at both the activity and the function of the brain, and on the communication between areas of the brain, and also the structure of the brain. So what's in our toolbox? These are the three most common imaging methodologies that are used to look at brain function and structure. Positron emission tomography, or PET, sorry, you might have heard of. And this is something which is not used in every center. Mostly it's in academic centers, although there are some many clinical applications now for PET. MRIs, magnetic resonance imaging, is quite common. Most of you have heard of this. And magnetic encephalography is something that is not as common as the other two. The machine looks kind of like a big hair dryer like this. You just sit under it, and it's a machine that picks up signals, magnetic signals that are generated because of the electrical activity of your nerve cells in your brain. So those are the three main techniques. Of course, there's also EEG, but I won't be talking about that today. So what can we measure? If we want to measure function, the function of the brain, we can use a type of MRI called functional MRI, or fMRI, or these positron emission tomography scans. We can also look at where drugs and different neurotransmitters act in the brain, and we can use PET to do that. We can look at the pathways and how information gets from one area to another, again using MRI. And if we want to look at both the gray matter and the white matter in the brain, again, there are structural MRI techniques to do that. So for the rest of my talk, I'm going to be concentrating on MRI. I won't be talking about PET. So let's talk about what you can do with functional MRI in a typical experiment. So this is just a little cartoon that I have drawn just to give you an idea of the different approaches that we have to using fMRI to understand what's going on in the brain. So the first thing that we can do is we can look at how treatments affect the brain. Now we can also look at, and we can do that in an individual person or across a group of individuals. We can also look at how the brain responds or changes over time when you learn to do something like playing an instrument. So this is, of course, scientifically interesting to understand how the brain shapes itself, but clinically it's also quite important because by understanding how the brain can change, even to something as simple as learning an instrument or training, we can understand the capacity of the brain to undergo change, both maladaptive and adaptive. And this gives us a hint as to what we can be aiming for in clinical studies when we try to reverse or change a brain that's not working well. Okay, now how do we find out that a brain is not working well? Well, we can do a bunch of experiments in a bunch of patients, and we can compare how they respond in an experimental situation to people who are healthy. And this is the typical thing that is done in brain imaging studies. We compare these two things. Now, how do we figure out then how there are abnormalities in all patients that are suffering from a particular disease or condition? So the trick is to try to figure out how to take an experimental sample of patients and healthy subjects and try to then make an inference to what's going on in the entire real population. And that's a difficult thing to do because, as I mentioned with the example of getting a flu shot, people respond quite differently. So to understand this intersubject variability and all the different ways that patients can respond, we need to be able to take into account all that intersubject variability and get a big enough sample and a varied enough sample to have some meaning when we try to apply it to the real population. So this is what we're aiming for. Now, how do we do it? So this is just a cartoon to explain some of the terminology that I'll be talking about. When we do an experiment, we have obviously a group of subjects, and they may be healthy individuals or patients suffering from a disease or condition. And they may undergo one or more brain imaging sessions where they come in and they have their brain scanned. And within each session, we can acquire data called runs that manipulate a task or deliver a stimulus or do a number of different things. So each of these recording sessions is called a run. So what happens in a run? Well, within a single run, you collect data over a period of time from either a specific area of the brain or, as shown in this cartoon, the entire volume of the brain. And that could be quite powerful to look at how the entire brain is behaving all at once. And how do we do that? Well, we don't actually take a picture of the whole brain at once. What we're actually doing is getting data from slices of the brain. So in this cartoon, you're taking slices, these horizontal slices or axial slices. So you're getting a bunch of slices of the brain, and the data that you actually collect is actually coming from tiny, tiny little pixels in this slice. So just like your computer screen you're looking at now or your TV screen is not just one big picture, but is comprised of many, many thousands of tiny little pixels, so is this image of the brain. Now we call it a voxel instead of a pixel because in reality it's not a totally flat two-dimensional structure, but it has a thickness to it, so it's actually a three-dimensional structure. So to give you an example, typical experiment in my lab, which might run for 10 minutes, might acquire data every 2 seconds from the whole volume, and in each slice, these might be 1 by 1 by 1 millimeter cubed voxels, so that ends up translating into about 1.2 million voxels in the entire brain, and you're collecting information in each of those 1.2 million boxes of the brain every 2 seconds. So you can imagine how enormous that data capture is. Okay, so what do we do with this information? Let me just give you a little peek behind the curtain of how we analyze these data and what kind of information we can get. So just to step back a bit, what is fMRI? So you know, we all see these fancy pictures in the media and in publications with nice colored blobs in the brain. So what produces that? Now, functional MRI isn't directly measuring the electrical activity of the neuron. It's inferring what's happening in those neurons based on an indirect measure. And this indirect measure is created because when neurons are active, they need more oxygen. This is the oxygen is the food that the neurons need to survive. So when neurons are active, what actually happens is you have increased blood flow that goes to that area of the brain, and that gives you more oxygen. It also means that the active area of the brain is using up the oxygen, so you have a change in what's called deoxygenated hemoglobin. And it turns out that this deoxygenated hemoglobin affects magnetic field lines. So this is what is being picked up by an MRI machine. It's picking up these changes in the magnetic field lines, and that translates into a signal that we measure. And that signal is called the bold signal or blood oxygen level detection signal. So one thing to keep in mind is this whole signal and what we're picking up due to the blood response, or what we call the hemodynamic response, is actually slow. So to give you an example, as I said, we can look at the brain every 2 seconds, get data every 2 seconds. And as I said, it's the data captures over many slices. And if we think about what's happening, let's say, for example, in one little voxel of the brain over time, let's say we look here at time 0, where we're going to give some sort of stimulus to the subject. And let's say they're looking, they're doing something, and they do it very quickly, and oh, there's a flash, just missed it, went very fast. A little flash of activity might have happened over a couple milliseconds. So what is the signal that we're picking up using an MRI? Well, the signal actually looks something like that. So this signal is not over milliseconds, but actually seconds. So it's thousands of milliseconds, and actually about 10 seconds long. So this response is slow. So keep that in mind when you're thinking about how the brain responds, and when you're looking at publications, that what we're looking at is this blood response, which is actually a little bit sluggish. It follows behind the actual brain activity. Okay, so how do you get these pretty pictures that are color-coded, these little blobs? What these blobs really represent are statistical outcomes of looking at the statistical tests that we use over every voxel in the brain to compare the signal when a stimulus is delivered or the subject is doing a task compared to a control period. So we do look at the comparison between these levels of the signal and these levels of the signal. We do a statistical test, and the outcome of that test gets color-coded, usually using hot colors like yellow and red to indicate that there's an increase. And so this is what is being shown in these maps. Okay, so let me now just explain another tool that we have in our toolbox to understand how the brain is functioning, and this is something called functional connectivity. So if you just look at this little cartoon I drew, which is supposed to represent kind of 5 areas of the brain that have some sort of connections between them. If you think about this network of the brain working together towards some common cause or outcome, we would say that this network is functionally connected if it shows activity which looks similar and is synchronized. And it turns out that brain areas show very slow oscillations over time, like this. This is a time course of an area of the brain over 10 minutes. And if we look at these slow, actually this is 5 minutes, and if we look at these slow oscillations, this is somebody lying in the scanner. It's probably me, actually, lying in the scanner, actually at rest. So this is what's called resting state functional connectivity. The person's not doing anything overtly, just lying there resting, and this voxel of the brain is kind of oscillating slowly. And what functional connectivity analysis does is it looks throughout the whole brain for areas of the brain, other voxels of the brain that are doing the same thing. It's doing exactly the same thing. And when we find these areas that show this synchronous activity, we say that these areas show functional connectivity. So it's not to say that they have necessarily an anatomic direct connection, just that they're doing the same thing at the same time, and so we think of it as being part of a common network. Okay, so what do we know about functional connectivity? Well, it really all began not that long ago. Back in 2001, Marcus Raichle first described this kind of strange, at the time we thought it was strange, that there was a set of areas of the brain that seemed to be active at rest, but decreased when people were doing a task. So he called this a task negative network, and actually named it the default mode network. And it turned out that the areas of the brain that turn on or are task positive are actually anti-correlated to this task negative or default mode network. So the default mode network is thought to play a role in monitoring your environment, your internal environment, and then maybe is suppressed when you have to focus your attention elsewhere. And we'll see in a few minutes why this is important to understand pain. So in addition to this default mode network, there's a network called the salience network and another network called the sensory motor network, which have been well studied. And these three networks seem to play a major role in pain. And so we'll come back to that in a few minutes. But in general, these functional brain networks defined using functional connectivity, or intrinsic brain networks, as they're also known, have now been very well studied. There's probably about a dozen of them, and they can be easily found, as I said, with the person lying in a scanner at rest, not doing anything overtly, and just looking at the activity of their brain. Okay, now just to finish off talking about techniques, I want to mention some techniques that comprise anatomic techniques, not only functional techniques. So we've all seen, again, these very beautiful pictures of the brain with brain connections going from one area of the brain to another. So there are ways that we can use MRI, namely a technique called diffusion tensor imaging, and it's able to measure connections from one area of the brain to another and to quantify that. Now we can also use diffusion tensor imaging to do something more quantitative, which is to look at the integrity of these white matter connections, to look at whether they're organized or disorganized, or maybe have an abnormality that may be linked to inflammation or edema, or perhaps demyelination, as may be the case in many diseases. So I won't go through the details of how we do that, but there are many methods that we can use to make these measurements. Okay, so let me now move on to talk about how we've been able to use these approaches to understand the brain and to understand pain in the brain. So I just want to start off by saying that when I first started doing brain imaging, what I noticed right away when I was collaborating with many of my clinical colleagues is that there are several chronic pain conditions in which the patients are not, it's not well understood, and many of these chronic pain conditions have been labeled as being idiopathic or functional because they can't seem to find any organic reason for the pain. And so this creates quite a stigma, and in many cases puts almost a blame on the patient, and really leads to a lot of stress and problems for the patient, for their work situation, their family members, and everybody involved. So having this kind of term idiopathic, I think, is a real problem, and I think we should find a different way about thinking about these conditions in which we don't understand a cause, because certainly there must be a cause. We just need to find it and study it and try to understand it. So the terminology is really one in which I think should be replaced with something else. So let me give you an example of that. We, in my lab, have been collaborating with many of our clinical colleagues, both neurosurgical colleagues and dentists, to look at a number of conditions. I'm going to mention two right now: trigeminal neuralgia and TMD, temporal mandibular disorder, so two orofacial pain conditions. And in both of these conditions, you have subsets of patients which are deemed to have idiopathic chronic pain because they can't find the cause. Now, in the case of trigeminal neuralgia, there has been a long-standing view that there is a vascular compression of the trigeminal nerve root, and that is underlying the patient's pain. The problem is with kind of a normal MRI or X-ray and type of clinical scan, you often don't see any sort of compression, even though the patient might be experiencing a lot of pain. And conversely, there are cases where there is compression of the trigeminal nerve root, but actually the patients are totally asymptomatic and they may not be experiencing any pain at all. So there's a disconnect between what you can see with kind of standard clinical scans and whether there is pain. And likewise, with patients who have been deemed to have idiopathic TMD, because you can't actually see anything wrong in the temporal mandibular joint that would cause pain. In fact, some people still have pain nevertheless. So we have used structural imaging, and I won't go into the details of it, but we have a number of publications in both these populations in which we were able to image, using structural imaging and diffusion tensor imaging, the trigeminal nerve, and we absolutely found very striking abnormalities in those nerves of these patients. So this is likely the cause or a major contributor to their pain. And so this just kind of hammers home the point that there are many conditions in which patients are deemed to have some sort of idiopathic pain or pain that people can't understand, but it's a matter of us developing the technology to find where the pathology is, and we're on the right track with many of these imaging approaches. Okay, the other abnormality that we have seen in patients has to do with functional connectivity. So this result is taken from a study by my student Casey Hemington, who found that patients who have pain associated with ankylosing spondylitis have an abnormality in how this default mode network communicates with the salience network. So normally, the default mode network and the salience network show anti-correlations, just like the slide I showed you a few minutes ago. But in these patients with chronic pain, that normal anti-correlation is attenuated and is hardly viewable. So this is just an example to show you that we can use both structural imaging and here functional connectivity imaging to find abnormalities in patients in which we don't really understand why they have pain. What Casey also found is that some of these abnormalities in the functional connectivity was related to how severe the pain was and how severe their actual disease was in terms of how it was affecting their activity of daily living. Okay, so in my the last part of this talk, what I want to highlight is the issue that I raised at the very beginning of this webinar, which is we need to understand the intersubject variability in both chronic pain and acute pain to understand how we can treat people optimally and kind of tailor treatments to each individual person. So I'm going to talk about individuality. And what we see when we look at healthy individuals is there's vast intersubject variability across many domains. So one domain is just the various measures of pain sensitivity, pain threshold, tolerance, habituation, and temporal summation. So I won't go into the details of this, but we know that there is a vast intersubject variability in how people tolerate pain, how they perceive a set stimulus. And in our lab, we've shown that there is a correlation between some of these measures, in this case, thresholds for heat pain and cold pain, and how thick the cortex is in some areas of the brain. This is the primary somatosensory cortex. So we see the beginnings here of hints that there is an underlying neural underpinning of this intersubject variability in pain sensitivity. And another study that was done by a former student in my lab, Javeria Hashmi, showed that people also differ in how they adapt to prolonged stimuli, let's say 40-second stimuli, and stimuli that are repeated. And what we found in this study is that there was a large intersubject variability. In this case, it was really based on sex. So women seem to actually have a dramatic adaptation to a prolonged stimulus and a repeated stimulus, where men have this sustained response to that same stimulus. And in a follow-up study that we did in our lab looking at connectivity, we found that men and women differed in how their brain was connected to the areas of the brain that are known to mediate pain modulation. So there is, again, an underpinning, a neural underpinning to why perhaps people adapt and habituate to stimuli differently. Okay, so there are a lot of complexities to the pain experience, both in terms of how we sense pain and how we modulate and cope with pain. And there are a vast number of brain areas and networks that underlie these things. So I've just shown you some examples related to kind of sensing and modulation. We've also looked at something called temporal summation of pain. Temporal summation of pain is a situation in which if you give repeated stimuli, let's say 10 stimuli, you'll tend to feel that stimulus as more and more painful over time. And if you use this as in an experimental way, you can kind of test how your NMDA system is working in your brain, because it's related to that. So in a recent study done by Josh Cheng in my lab, we looked at the intersubject variability of temporal summation, and this is across 70 individuals, and we saw that people had a different amount of temporal summation of pain. And it turns out that this fits into this concept that David Yarnitsky speaks about in terms of a nociception spectrum, and that is that there are people which seem to show a lot of this temporal summation and maybe also have less effective pain modulation systems, and he calls these people pro-nociceptive. And then people who actually don't show as much temporal summation and have maybe more efficient modulation mechanisms, and he calls these people anti-nociceptive. So what Josh did in my lab is he actually looked at whether or not there was a link between how the brain was organized and this temporal summation of pain behavior. And what he found is that people had a different balance of how strongly connected their sensing pathway was, or the ascending functional connectivity, versus their modulation pathway. And this seemed to relate quite strongly to the amount of temporal summation that they showed. So there was a link, and this functional connectivity seemed to predict this temporal summation of pain behavior. Okay, so I just want to mention a few other aspects of this intersubject variability, and this pertains to what we call in our lab the AP phenotype. And this is how people prioritize either doing a cognitive task, attending to that task, or dealing with pain. And we all have to cope with many things at the same time, and in some chronic pain patients, the pain interferes with them performing cognitive tasks, but actually in other situations distraction can be analgesic. So how do we balance these two things out? Well, over a series of studies that started with Dave Seminowitz's work in my lab in 2004, and then more recent studies by Natalie Erpelding and Aaron Kusi in my lab, we found two types of people. We call one type P-type because these people tend to attend to pain more when they're trying to do a task, and their performance on the task is reduced. But A-type individuals, or attention dominates, actually do the task quite well and can actually even improve their task performance when we're also delivering painful stimuli to them. So they can kind of put pain out of their mind and do the task better. And we have done a series of experiments that have found brain underpinnings in a variety of networks that seem to relate to whether people are A-type or P-type. Another more recent approach that we've looked at in the lab is to look at variability. And it turns out that in studies that have looked at aging, they have come up with the concept that variability in how your brain responds to various tasks can actually indicate brain health, and so it's a good thing. And so in our lab, Anton Rogachev has done a study which showed that that the higher brain variability, the more likely you were to be kind of an A-type person and do well on a cognitive task versus these people. So, so we have found a lot of underpinnings of this AP type. Now, in another approach to understand this kind of intersubject variability in how you deal with pain comes from a series of studies by Aaron Kusi when he was a student in my lab to look at mind wandering. And so what we did is we did a series of studies to look at how people attend to pain or mind wander from pain. And again, we found kind of a spectrum of people, and at one end, people with high intrinsic attention to pain, they actually were engaged in the pain, but other people that had low attention to pain, they mind wandered away from pain quite a bit. Now, are their brains different? Well, it turns out, and this becomes kind of complex, so I just suggest if you want to understand the details, you can go to our primary paper. But basically, we found that the brain does a variety of things to tune out pain when you're mind wandering. And what it does has to do with the salience network, the default mode network, and of course this descending pain modulation pathway. And so basically when you're tending to pain, there is a change in the activity of these areas, but when you mind wander from pain, then some of these areas kind of go back to similar to where they were at rest when there's no pain. So the nuance of all this, though, is that the connections between the brain in terms of functional connections, functional connectivity, is not static, but actually can change on a moment-by-moment basis. So people have now been looking at what's called dynamic functional connectivity. So they don't just look at the synchronization between two areas over, let's say, 10 minutes or 5 minutes while you're lying in a scanner, but they look at how variable those connections are, how dynamic those connections are. So in our lab, when Aaron and I continued looking at this question, we found out that people who were mind wanderers, able to kind of divert their attention away from pain, not only had a strong connection to their pain modulation pathway, but actually that connection was quite variable or dynamic. So we've called this system the dynamic pain connectome to capture that, that concept. Now, the last thing I want to mention, the last aspect of variability that we need to think about is personality and how we're able to modify our behavior. Now, I don't have time to go into all the details, but there are many factors that play into this that lead to these various responses of the pain varying. So, and we found areas in the brain that varied or were modulated differently if you had a different personality dimension. So the bottom line then is, we get back to the question, is the brain hardwired for pain? I think what we know now is that there are a lot of individual variations, but there's also moment-by-moment variations, and we know that we can actually change the brain, we can change the wiring of the brain, and the question is, can we then use what we know to target chronic pain and to target certain treatments with pain. So the real question is, can we predict treatment success? And that will allow us, if we understand a person before they undergo a treatment, how their brain is functioning, what kind of grouping they fall into, and whether or not a particular treatment then would be more effective for them than for some other people. And so this is all leading to this concept of personalized pain management, so that when a patient comes in, we can maybe do a lot of tests of the brain, but also behavioral tests. And by understanding how their brain is responding and how they behave to certain stimuli, we can direct their treatment to a certain option that might be targeting that particular system. So with that kind of approach, we're hoping to heal people's brains on a personal level, and that will give better pain relief with fewer side effects, fewer treatments to try before we hit upon something that's going to work. And of course, the ultimate outcome is to reduce societal costs, improve quality of life, and at the end of the day, people who are suffering tremendously, and even to the point of wanting to end their life, will want to live their life. So with that, I'm going to end the webinar, but I'm just going to mention for people who want to read up on any of the concepts in greater detail, we have a review on this dynamic pain connectome. You can read about that. We have another review recently that talks about the dynamics in general. Read about that. There's a related model that I think is important to understand how the brain is encoding pain. That came out last year. And then finally, some of the concepts I talked about and some of the cartoons I showed was from this TED Ed animation, which is now seen by over 1 million people, and that that will give you more information if you want to review that. And with that, I'd like to thank all the people in my lab over the years who've contributed to our work, and the funding agencies that have kindly supported my lab's work over the years. And with that, I would welcome any questions or comments.
Wonderful. Thank you very much, Karen. That was a really good presentation, and full of so much information that you also made very accessible, even though to some of us we might have been like, I don't know what's happening here. Very good. We don't have any questions submitted just yet, but I was curious, the last part where you were talking about the personalized pain care, I mean, you guys are researching it now. I mean, what's the likely timeline? I mean, is there any idea of when that is going to be an option for people
Yes.
on a more general level?
Yeah, absolutely. So I didn't show the slides, but we have data in our lab now that, and other people also in other labs, pain imaging labs, that have definitely shown that there are some baseline or pre-treatment indicators of treatment success. So a lot of people are using modeling and machine learning and other approaches to try to find predictors of treatment success, and even predictors. There is a lab in the States, Vanya Karin's lab, who has some very nice data showing the kind of experimental baseline levels that predicted whether people respond to, let's say, placebo. And they can predict with pretty high accuracy that subsequent response. And in some, we haven't published some of our data yet, so I can't talk specifically about it, but we've certainly found, at least at the group level, indicators, certain abnormalities that that reverse, for instance, with treatment in some patients, but not in others. So if we can find those before giving a treatment, if that indicator is there, then we, it's a better likelihood that patient will respond. So this is already starting to come out. It's obviously in the experimental phase. It's not in the clinic yet, and it's using relatively small groups of patients. So it's not ready for prime time to be rolled out into the clinic. But I'm quite hopeful that this is really coming, and it just requires a large effort worldwide to get more data.
That's wonderful. That's very hopeful and very fascinating as well.
Yeah, and I have to say this, this approach of personalized medicine, a decade ago this was, kind of science fiction, but now this is happening in all aspects of medicine. I mean, we know for many years now that even in cancer treatment, it's become personalized. Certain chemotherapy agents are chosen based on certain markers that a patient might have. Every aspect of medicine really is moving in this direction to try to come up with markers to personalize treatment.
Wonderful. All right. Is there research on the ability to change the dynamic pain connectome?
So that's a great question. Actually, we coined the term dynamic pain connectome just a year ago, so it's catching on. So things haven't been placed in that context just yet, but for sure we have seen, for instance, people who are studying cognitive behavioral therapy. That is one way of perhaps changing this connectome, and as we know, that works for some people very, very well, and for other people not at all. So again, if we can find out ahead of time which brain is more malleable to that particular kind of treatment, then we can save a lot of time and money in those patients. But yeah, certainly. These are questions we're asking in our lab about. I mean, people have been asking this question for years. They just haven't been using this particular terminology.
All right. And are you able to map the brain after a pain regime has started to see improvement?
Oh yes, absolutely. So we've been doing these studies for a number of years. These are hard. These longitudinal studies are quite hard because it involves bringing a patient in to have a lot of testing done before they start a treatment, and then following them over time. That's pretty difficult for a patient in terms of their time commitment. But we have been running and are currently running several studies that is looking at that. We did have a study we published last year that was focused on patients who had peripheral nerve injury, and we did follow them over time, and it was a small study, but did see a reversal of some brain abnormalities as over time as their pain was getting better. So this is something that we're doing.
All right. Someone asking, were the gender differences that you noted significant for both chronic versus acute pain?
Yeah, good question. So the example I gave you, that came from a study in healthy individuals and not in patients with chronic pain. Everything we're doing now is including looking at sex differences in chronic pain, but we don't have the data yet to show that. But we are studying some chronic pain conditions which are more predominant in one sex than the other, to try to see if there's a reason for this sex dominance in a particular condition in terms of brain wiring, and in conditions where you have chronic pain in both sexes, we're starting to look for those sex differences yet. So that information will be coming out in the not too distant future.
Wonderful. And what can we do to facilitate the transition from experimental stage to clinical use?
Yeah, that's a great question. Well, of course, funding is number one. I guess every scientist would say that. You know, we have been working in a very difficult financial situation in the last few years in terms of funding. So having increased funding would certainly allow us to do much more on a larger scale. I am very pleased that I'm part of the CIHR SPORE, which is Strategy for Patient-Oriented Research, in a group called the Chronic Pain Network. So some of my work is being funded there, and hopefully that will allow us to take things to the next level. The real one of the hardships and difficulties is in doing these studies is being able to do the studies in a large number of patients, and there's a lot of reasons why that's difficult. I think to have greater engagement of clinicians that aren't even necessarily engaged in academic research, but just are willing to give up some of their time to help us recruit patients and engage their patients in these studies, that would help tremendously, because it is hard to get a large enough group of patients to do, for instance, to look at sex differences. You know, you need to double your sample to do that kind of analysis. So we do need to engage as many clinics as possible in these studies.
And is there information on your website about how clinics can be in contact with you in order to do that? Because you're in Toronto, that's correct?
I'm in Toronto. One of the things that our network grant is aiming to do, of course, is to link groups across the country. So, you know, we are hoping to be able to do studies where, you know, if labs in different cities can collect some data and then the data comes to me where I can put it in a common database, we'd be able to do that. So that's something, you know, over the next 5 years we're working towards. That you know, takes a lot of time and effort, but that's what we're hoping to do. But certainly, if people want to email me, if they want to know more, I encourage and welcome anybody to email me to see how they can get involved.
Wonderful. And then, is there a neuroplasticity to the glia, and can gliosis be regenerated?
That's a wonderful question. Glad somebody raised the issue of glia, because your brain has 10 times more glia than it does neurons, and it's great that over the last decade the importance of glia has really come to the forefront, and I do collaborate with people who study glia, and in many instances I think understanding glia and neuroinflammation and those sorts of processes are really going to help us. And in fact, the glia system is one system which now we do know that there are some sex differences, and that's coming out of Mike Salter's lab and his colleagues. So I do talk to and collaborate with glia experts, and I think that is an emerging theme.
Wonderful. And then one more question. Are you able to differentiate the source of pain from two concurrent conditions?
That's a good question. Well, I think as we get more information about, each of those conditions separately, then we may be able to dissect that apart. But of course, that's a tricky situation because those two concurrent problems might interact with each other, so that it's not just pure sum of the two. But this is actually a very important question, because as we try to study certain kinds of patients that are older, certainly over the age of 50 or 60, you get into the situation where people do have multiple issues and multiple diseases and disorders. So and that does make looking at one in isolation of the other very, very tricky. But all I can say is, we hope to study the individual conditions so that we can then, try to make that separation later on.
Wonderful. All right. Thank you very much, Deborah, for the presentation and for all of the great information. I'm really pleased to have you here with us today.
Thank you. Thank you to all the audience online around the country and elsewhere.